The air filling your lungs right now is not some ancient, neutral background feature of this planet. It was made, manufactured, molecule by molecule, over geological timescales so vast they resist imagination, by organisms so small you could fit millions of them on the period at the end of this sentence. Without them, Earth would have remained exactly what it started as, a scalding, poisonous rock hurtling through space, hostile to every form of life we have ever known. The oxygen you are breathing, the nitrogen buffering every breath, the stable temperatures that allow liquid water to exist on the surface, none of it was given, all of it was earned. Through the slow, relentless chemistry of bacteria that had no plan, no goal, and no awareness of what they were building, they were simply surviving. And in doing so, they transformed a toxic wasteland into the only known home of complex life in the universe.
If you find yourself wanting to understand how something so small could rewrite the fate of an entire planet, hit subscribe. This channel exists precisely for questions like this one. Now, to understand what bacteria actually did, you first have to understand what they inherited, what Earth looked like before life got its hands on it. And the answer is something that should feel deeply wrong to any modern intuition about what a living world is supposed to be. Cast your mind back 4.5 billion years. The planet exists, but only just. The solar system is barely settled, and Earth is still being bombarded from all sides by asteroids and planetesimals left over from the formation of the Sun. The surface is not solid in any meaningful sense. Vast oceans of molten rock pulse and surge beneath a thin, brittle crust that cracks and reseals constantly. Temperatures at the surface hover at levels where most minerals simply cannot maintain a stable crystal structure. The sky above this churning landscape is not blue. It is not even close. The atmosphere at this stage is a dense, choking mixture of carbon dioxide, methane, hydrogen sulfide, ammonia, and water vapor, belched continuously from volcanoes that cover the surface far more extensively than any volcanic region that exists today. There is no free oxygen, not a single molecule of it in the air. The very concept of breathing, as animals understand it, would be instantaneously lethal here. The young sun, counterintuitively, is also significantly dimmer than it is today, outputting roughly 70% of its current luminosity. And yet the surface of early Earth is not cold. The greenhouse effect generated by those dense concentrations of carbon dioxide and methane traps enough heat to keep the planet from freezing. This is not warmth you would find comfortable. The oceans that begin to condense from water vapor, delivered partly by the same bombardment of comets and icy asteroids that was simultaneously terrorizing the surface, are warm to hot, and chemically nothing like the oceans we sail across today. Those early oceans are rich in dissolved iron. They carry hydrogen sulfide in concentrations that would be acutely toxic to virtually every organism alive on Earth right now. The water is slightly acidic, saturated with carbon dioxide dissolved from the atmosphere above. There is no ozone layer. Ozone requires oxygen to form, and there is no oxygen, which means the full, unfiltered ultraviolet radiation of the sun strikes the surface of the ocean and every exposed piece of rock without any atmospheric barrier whatsoever. The ultraviolet flux reaching the ground is far more intense than anything modern life experiences. It would shred the DNA of any contemporary organism within moments of exposure. This is the planet. Molten rock, ammonia-tinged skies, iron-laden sulfurous oceans, ultraviolet radiation washing over everything. A chemist handed this description without knowing it referred to Earth, would likely classify it as uninhabitable, not marginal, not challenging, but fundamentally incompatible with the biochemistry of life as we know it. And yet, something happened here. Something that represents perhaps the single most improbable event in the 4.5 billion years of this planet's history. Life appeared. The question of how is one of the deepest open problems in all of science, and the honest answer is that we do not fully know.
What we do know is where the strongest evidence points, and it points downward, into the ocean, to places where the laws of chemistry are bent by heat and pressure into something almost alchemical. Along the floors of those early oceans, wherever tectonic activity drove superheated water up through cracks in the seafloor, there existed what are called hydrothermal vents. Two broad types have been identified as relevant to the origin of life, and they are chemically very different from each other. The high-temperature black smoker vents belch water at extreme temperatures, rich in sulfides and metals. The cooler alkaline vents, structures like the Lost City field discovered in the Atlantic Ocean, produce a different chemistry, warm, mineral-rich water percolating through rocks in a process that generates hydrogen gas and creates a natural proton gradient between the alkaline vent fluid and the more acidic surrounding ocean water. That gradient matters enormously. The fundamental engine of life as we know it, the mechanism by which every living cell generates usable energy, runs on exactly this kind of gradient. Protons moving across a membrane from high concentration to low, driving a molecular turbine called ATP synthase. The alkaline vent hypothesis, championed by biochemist Nick Lane and others, suggests that the first protocellular chemistry did not need to invent this gradient. It simply found one that already existed in the rock and water, in the porous mineral chambers of the vent itself, and began exploiting it. Within those mineral chambers, the chemistry necessary for self-replication could concentrate and iterate. RNA molecules, capable of both carrying information and catalyzing chemical reactions, are the most favored candidates for the first genetic material. In the warm, mineral-rich microenvironments of alkaline vents, the building blocks of RNA can form abiotically, meaning without any biological help. Certain clay minerals and iron-sulfur clusters act as catalysts, accelerating reactions that would otherwise occur too slowly to be relevant on any useful timescale. At some point, and this is the moment that changed everything, the moment science cannot yet pinpoint precisely, a molecule formed that could make imperfect copies of itself. Imperfect copies that were themselves capable of making imperfect copies. From that moment, evolution had its raw material. The entity scientists call LUCA, the last universal common ancestor, is not the first life that ever existed.
It is the common ancestor of all life that exists today.
Every bacterium, every archaeon, every plant and animal and fungus alive on this planet traces its lineage back to this one population of organisms. LUCA was not simple, exactly. Reconstruction of its likely genome, based on genes shared across all domains of life today, suggests it had a sophisticated metabolism, membrane proteins, ribosomes, and a DNA-based genome. It almost certainly lived near hydrothermal vents, and, crucially, it did not breathe oxygen. It could not. Oxygen was not available. LUCA and its descendants fed on what the volcanic earth provided in abundance: hydrogen, carbon dioxide, hydrogen sulfide, ammonia. These are the molecules a living thing can use when no oxygen is present. Reducing agents, electron donors, chemical gradients that release energy when exploited by the right enzymatic machinery. The first ecosystems on Earth were not lush and green. They were invisible. Microscopic communities clustered around hydrothermal vents and shallow coastal waters, running entirely on the chemistry of a toxic planet. From our perspective, they were eating poison. From theirs, they were simply eating. For hundreds of millions of years, this was all there was, an entirely anaerobic biosphere, a world of life where oxygen would have been, at best, an irrelevant trace gas, and at worst, a genuine chemical hazard. The oceans teemed with microbial life, but that life was invisible, leaving almost no fossils behind. The chemistry of the rocks tells us they were there. The isotopic signatures of sulfur and carbon in ancient sediments preserve the fingerprints of metabolisms that have not fundamentally changed in 3.5 billion years. But the organisms themselves left almost nothing we can hold in our hands. The world above the waterline remained blasted by radiation, sterile,
and almost entirely lifeless. And then, somewhere between 3 and 3.5 billion years ago, the exact date is still debated, with some evidence pushing it earlier, a particular group of bacteria did something that no life on Earth had ever done before. Something so chemically novel, so energetically powerful, and so consequential for the future of the planet that it deserves to be called the most important biological innovation in the history of this world. They learned to split water using sunlight. This is oxygenic photosynthesis, and the organisms that invented it were the cyanobacteria. To understand why this matters, consider what came before. Early forms of photosynthesis already existed. Microbial mats and purple and green sulfur bacteria had been harvesting light energy for a long time, using hydrogen sulfide or hydrogen gas as their electron donor. These are anoxygenic photosynthesizers. They use light. They build organic molecules.
But they do not release oxygen. They are limited by the availability of their electron source. Hydrogen sulfide, for instance, is not infinitely abundant. Water, on the other hand, covers most of the planet. It is essentially unlimited. And somewhere in the lineage that would become cyanobacteria, a molecular mechanism emerged, the protein complex called photosystem II, that could use the energy of sunlight to rip apart water molecules and extract electrons from them. The residue of this reaction, the byproduct that was simply expelled as metabolic waste, was oxygen gas. At first, this waste went nowhere visible. The early oceans were saturated with dissolved iron, and oxygen reacts with iron instantly and aggressively. Every molecule of oxygen that the early cyanobacteria produced was almost immediately absorbed by the ocean, reacting with dissolved iron to form iron oxides that sank to the sea floor. Layer upon layer of these iron oxides accumulated over hundreds of millions of years, building up the banded iron formations, those distinctive striped rocks, red and gray and red again, that geologists mine today as iron ore, and that record, in their very layering, the pulse of an invisible biological revolution. The iron in those rocks was pulled from ancient oceans by the earliest oxygen ever produced on Earth. For a remarkably long time, perhaps as much as 600 million years, the ocean simply absorbed the oxygen. Anytime cyanobacteria exhaled it, the iron consumed it before it could accumulate. But iron is not infinite either. Slowly, gradually, the iron in the shallow coastal seas was depleted, the rust settled, the sink shrank, and the oxygen began, for the first time in Earth's history, to accumulate in the water, and eventually to leak into the atmosphere above. What happened next was, from the perspective of the life that existed at that moment, an apocalypse. 2.4 billion years ago, give or take a few tens of millions of years, with some studies placing the precise onset slightly later, the concentration of atmospheric oxygen crossed a threshold. This is the Great Oxidation Event, sometimes called the oxygen catastrophe. And the name is not hyperbole. Oxygen is a ferociously reactive molecule. For the anaerobic organisms that had dominated Earth for well over a billion years, it was not a new resource, it was a poison. Oxygen attacks organic molecules, destroys enzymes, generates reactive oxygen species inside cells that rip apart membranes and shred genetic material. The microbes that had built every ecosystem on Earth had no defenses against it, because there had never been any need for such defenses. The ecological collapse was staggering. Isotopic evidence from ancient sulfate minerals suggests the total size of the biosphere may have shrunk by more than 80% during and after the Great Oxidation Event. Anaerobic organisms that had owned the planet for billions of years were pushed back to the margins, retreating into the oxygen-free refuges that still exist today. Deep ocean sediments, waterlogged soils, the guts of animals, the airless mud of wetlands. Those refuges are still full of their descendants, living almost exactly as their ancestors did before the catastrophe, waiting out a world that turned against them. The banded iron formations in the rock record are not merely a geological curiosity. Each red stripe is a layer of iron oxide, Iron that was pulled from the ocean by cyanobacterial oxygen, oxidized, and dropped to the sea floor. They are, in effect, a stone ledger of mass death. Billions of years of biological exhaust recorded in mineral layers, a monument to the slow drowning of an entire pre-oxygen world. When the iron ran out and the oxygen finally broke into the atmosphere, those stripes stop appearing. The ledger closes. Something irreversible has happened. Not everything died. The survivors fell into two categories. Those already occupying environments where oxygen could not reach, deep sediments, hydrothermal systems, anoxic basins, persisted in the pockets of an oxygen-free world that never fully disappeared. And a second group had, through evolutionary tinkering near cyanobacterial oxygen oases, already developed the machinery to detoxify reactive oxygen species and eventually to harness oxygen for respiration. Research published in recent years suggests that aerobic respiration, the ability to use oxygen to extract energy from food, evolved as early as 3 billion years ago, hundreds of millions of years before the Great Oxidation Event permanently flooded the atmosphere. These early aerobes likely lived close to cyanobacteria, consuming the small oxygen oases the photosynthesizers produced locally, and may have actually helped delay the Great Oxidation Event by absorbing oxygen before it could accumulate in the wider ocean. Among the survivors, cyanobacteria themselves were conspicuous. They had been producing oxygen and living adjacent to it for hundreds of millions of years. Their cells carried enzymes, catalase, superoxide dismutase, that neutralized reactive oxygen species before they could cause cellular damage. These enzymes are ancient, and their presence in virtually every aerobic organism alive today speaks to how fundamental the problem of oxygen toxicity was, and how early the solution was found. The geochemical fingerprint of the Great Oxidation Event is written into the rock record simultaneously in multiple ways. The disappearance of mass-independent sulfur isotope fractionation, a chemical signal that can only be produced in an oxygen-free atmosphere, marks the moment the atmosphere crossed its threshold with unusual precision. New minerals begin appearing, oxidized minerals, red beds, ancient soils called paleosols that carry the chemical signature of oxygen weathering, marking a planet whose surface chemistry has been fundamentally and irreversibly altered. A world that had been chemically stable for over 1 billion years was suddenly running a completely different set of reactions on its surface. And one of the immediate consequences of those new reactions was catastrophic in its own right. Oxygen does not only attack iron and organic molecules, it attacks methane. The early Earth atmosphere contained enormous quantities of methane, produced by the vast communities of methanogenic archaea that had been running global biogeochemistry for billions of years. Methane is a powerful greenhouse gas, and as long as it persisted in the atmosphere, it had been supplementing the warmth provided by an otherwise still dim young sun, keeping the planet above freezing. When oxygen arrived in the atmosphere, it began converting methane to carbon dioxide and water. The warming blanket that methane had provided was stripped away over geologically rapid timescales. The planet, exposed suddenly to a dimmer sun without its methane buffer, began to cool. It cooled dramatically, catastrophically. The result was the Huronian glaciation, a series of ice ages lasting from roughly 2.4 billion to 2.2 billion years ago, representing some of the most severe glacial episodes in Earth's history. Evidence suggests that ice may have extended to equatorial latitudes during at least some of these glaciations, effectively encasing the planet in a shell of ice from pole to pole. This is the phenomenon known as snowball Earth, and it was not the last time Earth would experience it. But this first iteration was directly caused by the chemistry of life rewriting the atmosphere. The albedo feedback accelerated the freeze. White ice reflects sunlight far more efficiently than dark ocean or land. So the advancing glaciers reduced the amount of solar energy the planet absorbed, which cooled it further, which drove the glaciers further still. Beneath the ice,
in the liquid water preserved by geothermal heat, in the thin photosynthetic communities clinging to whatever light filtered through the thinnest ice margins. Bacteria endured. Chemolithotrophs, organisms that draw energy from inorganic chemical reactions rather than sunlight, thrived in the permanent darkness of subglacial waters, feeding on hydrogen and sulfur compounds released by hydrothermal systems that had not stopped operating simply because the surface above them was frozen solid. Cyanobacteria clung to whatever surfaces allowed enough light through to sustain photosynthesis. Present-day analogues of these environments, subglacial lakes in Antarctica, permanent polar sea ice, contain living microbial mats, direct evidence that bacterial life can persist through conditions that render the surface entirely uninhabitable. Escape from the freeze came from geology. Volcanoes continued outgassing carbon dioxide throughout the glaciations, and with the land buried under ice, that carbon dioxide could not be removed from the atmosphere through normal silicate weathering. It accumulated until the greenhouse effect it generated finally overwhelmed the albedo feedback of the ice. The planet warmed, the ice melted, and the thaw released an enormous quantity of nutrients that had been locked under glacial sheets: phosphorus, iron, silica, minerals ground from deep crustal rocks by ice and held captive for millions of years. Released all at once into newly open ocean, they fertilized the biosphere on a planetary scale. Life exploded in response every time the ice retreated, each recovery leaving the planet slightly more oxygenated and slightly more hospitable to complex aerobic chemistry than it had been before the freeze. The nitrogen cycle, though, was running beneath all of this, and understanding the full architecture of what bacteria built requires following that chemistry carefully, because without it, none of the biological complexity that followed was chemically possible. The atmosphere is close to 80% nitrogen gas. Nitrogen is essential to life in a way that is almost impossible to overstate. Every amino acid, every protein, every molecule of DNA and RNA contains it. But nitrogen gas, two atoms triple bonded to each other, is chemically so stable it is essentially inert to most biological chemistry. It cannot be incorporated into organic molecules without first being broken apart and combined with hydrogen to produce ammonia. Only bacteria carry the enzyme nitrogenase capable of doing this. Specific groups of bacteria, including certain cyanobacteria and a wide range of free-living and symbiotic soil bacteria, use nitrogenase to cleave the triple bond of nitrogen gas and attach hydrogen atoms to the resulting nitrogen atoms, producing ammonia. The reaction is extraordinarily energetically demanding. Isotopic evidence from rocks dating back 3.2 billion years shows the signature of biological nitrogen fixation already operating in the Archean oceans. Researchers who reconstructed ancient nitrogenase enzymes from that deep in evolutionary history and synthesized them in the laboratory found that those ancient enzymes produced the same characteristic isotopic fractionation patterns as modern ones, meaning the core chemistry of nitrogen fixation has been conserved across more than 3 billion years of evolution. Without nitrogen fixation, the ocean's reactive nitrogen supply would have been limited to whatever small amounts lightning and ultraviolet photochemistry could produce abiotically. That input is real, but tiny. The biological nitrogen cycle delivers reactive nitrogen to ecosystems at rates that dwarf any abiotic source. The nitrogen in every muscle fiber in your body passed through a bacterial nitrogenase at some point in its history. The nitrogen cycle is not a one-way street. Once bacteria fix nitrogen into ammonia, other bacterial communities take that ammonia and oxidize it to nitrite, then nitrate, in a process called nitrification. And then yet another guild of bacteria, the denitrifiers, takes those nitrates and reduces them back to nitrogen gas, returning the element to the atmosphere and closing the loop. This cycling keeps the atmosphere in chemical balance and prevents reactive nitrogen from accumulating to toxic concentrations in the ocean. It was running, invisibly, relentlessly, long before any multicellular organism drew its first breath. The central irony of this story is that the same oxygen making aerobic life possible
is also the chemical enemy of the enzyme that builds aerobic life's molecular building blocks. Nitrogenase is irreversibly destroyed by oxygen. Its metal cluster, which breaks the nitrogen triple bond, is oxidized and rendered inactive on contact with it. Cyanobacteria that fix nitrogen solve this by dedicating specialized cells called heterocysts to the task, maintaining an oxygen-free interior within those cells while the surrounding photosynthetic cells pump oxygen out. Other nitrogen-fixing bacteria live in soil or root nodules, in anoxic microenvironments carefully maintained to protect the enzyme from the aerobic world outside. What bacteria built across those billions of years before complex life appeared was not just a collection of chemical reactions, it was infrastructure. Planetary-scale infrastructure, as real and as essential as any physical structure, maintained not by design, but by the accumulated consequence of individual metabolisms doing whatever kept each cell alive from one moment to the next. The atmosphere was the output of a biological machine that never intended to build anything at all, and that machine had already produced the precise chemical conditions that would make the next transformation possible. A transformation unlike anything that had come before, because it required not one organism inventing something new, but one organism being swallowed by another, and neither of them dying. Something happened roughly 2 billion years ago that evolution had never managed before and would never quite repeat. A cell, almost certainly an archaeon, a member of that ancient lineage of microbes that had survived the oxygen catastrophe in the deep, anoxic refuges of the ocean, engulfed a bacterium. Predatory engulfment is an ancient strategy, and countless cells had been swallowing other cells since the earliest days of microbial life. What was unusual was what happened next. The bacterium was not digested. It kept living. Inside the host cell, surrounded by the machinery of a foreign organism, the engulfed bacterium continued running its metabolism. And that metabolism, aerobic respiration, the ability to extract energy from organic molecules using oxygen, was something the host cell could not perform on its own. The host provided shelter, an internal chemical environment, and access to organic carbon. The bacterium provided energy, in quantities that dwarfed what the host could generate independently. Neither organism was the same afterward. Neither could return to what it had been before. Lynn Margulis understood this in a way that the scientific establishment of the 1960s was not prepared to accept. She was 28 years old when she circulated the manuscript that would eventually become her landmark 1967 paper. And she was working from a convergence of clues that others had noticed individually, but never assembled into a single, coherent claim. Mitochondria, the energy-generating structures inside every eukaryotic cell, had their own DNA. Their ribosomes resembled bacterial ribosomes far more closely than they resembled the ribosomes in the surrounding cell. Their inner membranes were structured like bacterial membranes. They divided by binary fission, the way bacteria divide, not by the elaborate choreography that eukaryotic cells use to copy their chromosomes. And they were the right size, exactly the right size, to be former free-living bacteria that had taken up permanent residence inside a host. Her paper was rejected by 15 journals before it was published. The core claim that the fundamental structures of complex cells were not invented by those cells, but were bacteria incorporated through endosymbiosis, struck most of her colleagues as too radical, too sweeping, too disruptive of the received view of how evolutionary novelty was supposed to arise. Evolution proceeded by gradual mutation and selection, the consensus held, not by one organism literally moving inside another and staying there permanently. The evidence was unambiguous, and it only accumulated over the following decades. When molecular sequencing became possible, the genetic relationships confirmed what Margulis had argued from structure alone. Mitochondrial DNA is unambiguously bacterial in origin, most closely related to a group called the alpha proteobacteria. Chloroplasts, the photosynthetic organelles of plant cells and algae, are equally unambiguous descendants of cyanobacteria,
the same lineage that triggered the Great Oxidation event and very nearly destroyed anaerobic life on Earth. The organisms that produced the oxygen catastrophe were eventually folded into the cells of eukaryotes and put to work powering the bodies of every plant that would ever grow. Stand at a microscope and look at a living plant cell, its chloroplasts moving slowly through the cytoplasm in the gentle circulation called cyclosis, each one a green, lens-shaped body about the size of a small bacterium. Each chloroplast still carries its own genome, a shrunken remnant of its original bacterial chromosome, encoding the proteins most critical to photosynthetic function. Over 1 billion years of living inside a host, most of its genes have been transferred to the host cell's nucleus, a slow gene-by-gene migration that has been directly observed in real time in laboratory experiments, as DNA escapes the organelle and integrates into the nuclear genome. The chloroplast exists in a state of exquisitely managed interdependence, replicated when the cell divides because the cell could not survive its loss. Endosymbiosis built an entirely new tier of biological organization. The eukaryotic cell, the cell with a nucleus, with mitochondria, with the capacity for internal membrane systems of startling complexity, could not have arisen any other way. The energetic mathematics of the problem are decisive. A bacterial cell can generate only a limited amount of energy because it can only pack so many energy-generating proteins into its single membrane. A eukaryotic cell with thousands of mitochondria, each one a former bacterium with its own dedicated membrane surface area, can generate energy at a per-gene rate that no prokaryote can approach. This energy surplus is what pays for complexity, for elaborate gene-regulatory networks, for the production of thousands of different protein types, for the maintenance of large genomes, for the construction of multiple cell types working in coordination. Every multicellular organism alive today, every plant, animal, fungus, and complex alga, is a direct beneficiary of the metabolic windfall that originated when one cell failed to digest another. Margulis spent the rest of her career extending the logic of endosymbiosis further than many of her colleagues were willing to follow, proposing that other cellular structures also had bacterial origins, and that symbiosis, not competition alone, was a primary driver of evolutionary innovation. Some of her later proposals remain contested. The core insight, though, that cooperation between organisms formerly separate, formerly enemies, could generate biological novelties that neither could achieve alone, stands as one of the transformative ideas in the history of biology. The emergence of eukaryotic life was not a gradual refinement of bacterial chemistry. It was a merger, and that merger made complex multicellular life possible. Complex multicellular life needed a surface to live on. For most of Earth's history, the surface above the waterline was not somewhere anything complex could survive. The ultraviolet radiation reaching the ground before the Great Oxidation event and the ozone layer it enabled was enough to damage DNA faster than any unprotected organism could repair it. Even after oxygen began accumulating in the atmosphere, the ozone layer was thin and variable, and its full development tracked the gradual rise in atmospheric oxygen concentrations across hundreds of millions of years. Ozone, 3 oxygen atoms bonded into a single molecule, forms in the stratosphere when ultraviolet radiation splits ordinary oxygen molecules into individual atoms, which then bond with intact oxygen molecules. The ozone layer depends directly on the concentration of atmospheric oxygen. The biosphere's photosynthetic output, cyanobacteria, algae, and eventually land plants, was ramping up through the Proterozoic, driving oxygen concentrations toward the levels at which a meaningful ozone shield could form. The two systems, biological and atmospheric, were in direct chemical conversation, each setting the conditions for the other. At current oxygen concentrations, the ozone layer sits primarily between 15 and 35 kilometers above the surface. From the ground, it is invisible and undetectable to human senses. From a research aircraft flying at the altitude of the lower stratosphere, its presence becomes measurable in the dramatic drop in ultraviolet irradiance below the layer. Above the ozone, the full solar UV spectrum arrives intact,
including the deep ultraviolet wavelengths most destructive to nucleic acids. Below it, those wavelengths are almost entirely absorbed. The layer sits between the living world and a radiation environment in which complex surface life could not persist. The ozone shield was never constructed. It appeared as a necessary atmospheric consequence of the cyanobacterial exhaust that had been accumulating since the Great Oxidation event. The protection that allows land plants to photosynthesize, that allows insects to navigate, that allows every terrestrial vertebrate to go about its life under open sky. All of it traces back to the metabolic waste of organisms that had no more awareness of what they were building than a fire has awareness of the warmth it produces. Below the ozone layer, on the land that the shield gradually made habitable, another bacterial revolution was unfolding.
The transformation of bare rock into soil is easy to underestimate because soil is so ordinary, so present, so apparently simple. Pick up a handful of it, and it looks like nothing in particular. Dark, crumbly, faintly organic in smell. That smell is geosmin, a compound produced by actinobacteria in the soil, detectable by the human nose at extraordinarily low concentrations. The nose can detect it because human ancestors needed to find water, and geosmin signals biological activity in soil, which signals moisture and fertility. The human sense of smell was partly shaped over millions of years of evolution to locate the presence of soil bacteria. Bare rock, exposed to the atmosphere, weathers slowly through wind, rain, and temperature cycling.
Biological weathering proceeds faster because bacteria and their associates, fungi and lichens, which are themselves symbioses between fungi and algae or cyanobacteria, produce acids and chelating agents that dissolve minerals far more efficiently than rainwater alone. Pioneer communities of cyanobacteria colonize bare rock surfaces, fixing nitrogen, weathering the mineral surface chemically, producing organic carbon that accumulates in the rock's interstices. As they die, their organic remains mix with the mineral particles they have helped free from the rock. Fungi extend their hyphae into the matrix, breaking down both mineral and organic material. The first recognizable soil forms. A soil scientist working with ancient cultivated soils, the agricultural soils of the Near East or China, farmed for thousands of years, will note that the fertility of those soils reflects the microbial communities within them. A gram of fertile topsoil contains somewhere between 100 million and 1 billion bacteria, belonging to thousands of species. Those bacteria decompose organic material, cycle nitrogen and phosphorus through forms that plant roots can absorb, suppress plant pathogens, and maintain the porous physical structure of the soil that allows water and air to penetrate. Sterilize that soil, and it becomes chemically inert. Plant roots placed in it cannot take up nitrogen or phosphorus efficiently, regardless of how much of those elements is physically present. The availability of nutrients to plants is not simply a matter of chemistry. It requires living mediation. The first land plants that colonized the terrestrial surface in the Ordovician, roughly 470 million years ago, did not do so alone. They brought their microbial partners with them. Mycorrhizal fungi, which themselves depend on bacterial communities in the soil, were almost certainly colonizing early plant root systems from the beginning of terrestrial plant evolution. The fossil record shows fungal structures inside the cells of some of the earliest land plant fossils, in an intimate association that has persisted in the vast majority of plant lineages to the present day. The plants provided the fungi with organic carbon from photosynthesis. The fungi extended the effective reach of the roots into the soil matrix, accessing mineral nutrients, phosphorus especially, that the roots could not have reached on their own. Both relied on bacterial communities to cycle nitrogen and maintain the chemical environment of the soil. The colonization of land was not a single biological event. It was a coordinated movement of an entire ecological network. Plants, fungi, and bacteria, each component already in relationship with the others for millions of years in aquatic environments. The land did not receive life. It received an ecosystem. Today, the full scale of what bacteria maintain on this planet is something that climate scientists and microbiologists are still mapping. Every major biogeochemical cycle, Carbon, nitrogen, sulfur, phosphorus, iron passes through microbial metabolism at critical steps. The carbon dioxide concentration of the atmosphere, which determines global temperatures, is regulated in part by the photosynthetic uptake and respiratory release of carbon by microbial communities in the ocean. Marine cyanobacteria, particularly the tiny but extraordinarily abundant Prochlorococcus, discovered only in 1988, account for a substantial fraction of all oceanic primary production. Prochlorococcus cells are so small and so numerous that they collectively contain more carbon than all the fish in the ocean combined. They are the base of food chains, supporting every marine ecosystem on Earth. A climate scientist working with biogeochemical models of the ocean encounters bacteria not as incidental actors, but as rate-limiting parameters in the equations.
The speed at which carbon sinks from the surface ocean to the deep, the biological pump, which moves carbon from the atmosphere into long-term storage in deep sediments, depends on the activity of bacterial communities decomposing sinking organic material. The efficiency of nitrogen fixation in the open ocean determines how much of the surface can support photosynthesis. The production of dimethyl sulfide by marine bacteria and their algal associates generates cloud condensation nuclei over the open ocean, influencing cloud cover, albedo, and regional climate. The atmosphere and the ocean and the land surface are not physical systems that happen to have life on them. They are systems whose current state is a direct consequence of ongoing microbial metabolism. Consider what stops if that metabolism pauses. Without decomposer bacteria, organic material accumulates without being broken down. Nitrogen fixation halts. The supply of reactive nitrogen to plant roots and oceanic phytoplankton collapses. Photosynthesis falters, as nitrogen-starved organisms cannot synthesize the chlorophyll and enzymes needed to capture light. The biological pump in the oceans ceases, and carbon that would have been sequestered in deep sediments returns to the atmosphere. Without microbial communities maintaining soil structure, terrestrial soils lose their porosity and fertility. The cascade is total. There is no version of Earth's current climate, current atmosphere, current ocean chemistry, or current soil fertility that persists without the bacteria actively maintaining it. The oxygen entering your lungs right now was released by a cyanobacterium in the ocean within the past few years. The supply is continuously renewed by photosynthesizers who have been performing this chemistry since before the Cambrian. The nitrogen in the proteins of your muscles passed through a bacterial nitrogenase. The carbon in your bones was drawn from the atmosphere by a photosynthesizer and passed through a food chain that bacteria decomposed and rebuilt at every step. Your mitochondria, generating the energy that powers every thought and movement, are the direct descendants of bacteria swallowed and retained 2 billion years ago. Every human being is, at the most fundamental level of biological accounting, a temporary confederation of ancient bacterial chemistry, walking under a sky that bacteria oxygenated, eating food grown in soil that bacteria built. The entire human project, civilization, agriculture, technology, every cultural achievement across the full span of recorded history has been conducted within a biosphere that bacteria created and that bacteria maintain. Put it this way, you are not an organism living on a planet. You are a cell living inside an organism, one that has been breathing, cycling, and self-regulating for 3 billion years, and that was ancient before animals were even a possibility. The bacteria did not build this world for us. They built it for themselves. We are simply one of the more elaborate structures that their chemistry made room for. If this kind of deep perspective on the world you live in is what you're after, subscribing to this channel will make sure you don't miss what comes next.